So we're on to the second part of our antibiotics and resistance mechanisms lecture. And in this lecture, we're going to talk about inhibition of folate synthesis, a different group of antimicrobial agents. So the folic acid pathway in bacteria is essential for DNA biosynthesis, and some of the antimicrobial agents will block this pathway. So examples of some of these agents are sulfamethoxazole, trimethoprim. They're commonly used together. So sulfamethoxazole, trimethoprim are many times combined. These are completely synthetic, so they are antimicrobial agents. They are not antibiotics, which are natural. When they're combined together, which I already said they're commonly combined, they're broad spectrum, and that's why many times they're made together as it's one pill, but they have both antimicrobial agents in it. And the sulfamethoxazole trimethoprim combo are commonly used for urinary tract infections that are caused by our Enterobacteriaceae members. And by now, you should be thinking of all the different organisms that can cause urinary tract infections. What's the number one organism that causes urinary tract infections? Come on, I know you know it. It's E. coli, number one. There are many other organisms that cause urinary tract infections. Proteus can cause urinary tract infections. Klebsiella, Staphylococcus aureus can cause urinary tract infections. Staphylococcus saprophyticus, our organism that commonly causes urinary tract infections in sexually active females. There's all sorts of organisms. So whenever at this point we're talking about a type of infection, you should be able to think of all the different agents and which ones are the most common, which ones are you as a clinical laboratory scientist most likely to come across when you're working in clinical micro. Okay, back to antibiotics. The next group of antimicrobial agents that we are going to discuss are those that target the cytoplasmic membrane. These antimicrobial agents will alter the cytoplasmic membrane. What happens when you alter the cytoplasmic membrane of bacteria is you get leakage of the cell. Examples of these types of antimicrobial agents that will attack the cytoplasmic membrane are polymyxin B, this is an antimicrobial agent commonly used for Pseudomonas infections. Speaking of urinary tract infections, Pseudomonas aeruginosa can cause urinary tract infections and causes many different types of infections, opportunistic organism. Amphotericin B is useful for fungal infections. You will be covering the fungal organisms next semester in microbiology 2, so you won't have to worry about those this semester, thank goodness. Nystatin is useful to treat yeast infections, and yeast is a type of fungus, your fungal organisms. You have yeasts and molds. So again, you won't talk about yeast until next semester for those of you that are taking Micro 2. And then you have your imidazoles, such as miconazole, ketoconazole, fluconazole. And again, these are antifungal agents. Now we're going to talk a little bit, we're just briefly discussing these, the antimicrobial agents that inhibit DNA synthesis. These include the quinolones and the fluoroquinolones. And some examples of the quinolones are norfloxacin, ciprofloxacin, levofloxacin. These are completely synthetic. They are antimicrobial agents. They affect the enzyme DNA gyrase. So for those of you that are that have taken in the past a molecular biology course, any of you taking molecular biology this semester or molecular diagnostics should be very much aware of the enzyme DNA gyrase. So DNA gyrase affects the helical structure of DNA in our cells. These are broad spectrum antibiotics or antimicrobial agents, I should be more correct, and they are very powerful bactericidal. So cidal means they kill the prokaryotic cell. Metronidazole is another example of an antimicrobial agent that inhibits DNA synthesis. Examples are flavodoxin and ferredoxin, and these are activated by bacterial proteins that are found in both microaerophilic as well as anaerobic bacteria. So what these antimicrobial agents do is they nick the bacterial DNA. And these agents are optimally active in low redox potential, so low oxygen environments, which is why they are going to be more effective with your anaerobes or microaerophilic organisms because they're in an atmosphere of lower oxygen potential. The next group of antimicrobial agents are those that interfere with DNA transcription. And these agents will prevent the synthesis of messenger RNA from DNA. So again, for those of you that are taking molecular diagnostics, we went through this a lot this semester. You start out with your DNA. From your DNA, it's transcribed to messenger RNA, and from messenger RNA, it is translated into a protein. Every single solitary cell needs proteins in order to function. So if you're preventing the synthesis of messenger RNA, you're ultimately blocking the cell's ability to generate any proteins. So examples of antimicrobial agents that block transcription by preventing messenger RNA synthesis are the rifampins. Rifampins inhibit RNA polymerase. These are broad spectrum antibiotics, and they're commonly used to treat Mycobacterium tuberculosis infections, but they can also be used for other infections caused by staph, enterococcus, haemophilus, strep pneumoniae. The next group interfere with messenger RNA translation. So these inhibit protein synthesis. So they target protein synthesis and metabolism. They prevent bacteria from synthesizing structural proteins and enzymes. The problem with some of these agents is they can be toxic. Now, the agents that will block translation, they do this in many different ways. They can alter the ribosome because the ribosome is what comes in, attaches to the messenger RNA, and moves down the messenger RNA to translate those triplet codons. So if they alter the ribosome, you're not going to get translation. Some of these antimicrobial agents will bind to the 30S subunit of the ribosome. Some of them will bind to the 50S subunit of the ribosome. You don't need to know which ones bind irreversibly, which ones bind reversibly. That's a little too specific. So just know that some agents block translation, protein synthesis. So those that block protein synthesis are the aminoglycosides such as streptomycin, kanamycin, amikacin. These are broad spectrum agents. They can be resistant to aminoglycosides, and anaerobes cannot absorb these types of agents. They're naturally resistant. There are some agents such as tetracycline, monocycline, and doxycycline. You don't have to know the specifics of which one binds to what. I mean, I think for micro, I think it's just important to know that there are different types of antimicrobial agents, what the different categories are, and what their target is, and that's it. You don't need to know their exact mechanism. If this was a pharmacology course, certainly you would definitely need to know that. Those that bind the 50S, again, you don't have to know specifically, just that they affect protein synthesis, the lincomycin and clindamycin. The macrolides, such as erythromycin, claromycin, and azithromycin, are broad spectrum. And somewhat newer, but there's always new antimicrobial agents being developed, so it's very difficult, this field, to stay on top of. Targeting protein synthesis, there's linezolid, there's degacycline, there's all sorts of new agents. Now, there are also antimicrobial agents that are very specific. They're only treated for certain organisms. So one group are the anti-mycobacterial agents, and these include rifampin, which we already mentioned. Rifampin is also sometimes referred to as rifampicin, isoniazid, ethambutol, pyrazinamide, streptomycin. So this group of antimicrobial agents, some of them can be used to treat other infections, but commonly these are the agents that are going to be used to treat a TB patient. We're now going to move on to the third part of this lecture.